The very last thing we got, everybody, is cell signaling. Okay, cell signaling. Okay, yeah, it's still working. All right. We need to have receptors that to tell the cells to do stuff, generally. We can also have gap junctions as well, all right? We see those, we're going to see those in the heart. We're going to see those in smooth muscle as well, okay? Gap junctions allow adjacent cells to quickly move ions or regulatory molecules from one cell to the next. Okay, there's a hole that exists in between them, so they can go easily from one into the next. We also have that thing that I mentioned earlier with paracrine secretion. Parallel signaling. Okay. Paracrine signaling is only local. Okay, because you may then, some people may think paracrine secretions are going to be able to get into the fluids and then get to the adjacent cells, which they do. But then if they're in the fluids, that means the blood can take them in, transport them around. And that is not necessarily the case. All right? They stay very localized. Paracrine signaling is only good right next to where they get released from, because that's where the greatest concentration is. As soon as they get whisked away by blood, they are now in a hell of a lot lesser concentration than right where they got released from, where the greatest concentration is. All right. So that's why a paracrine signal is a localized event. And that's great, okay? Because we see a lot of this in terms of hemostasis. Okay? When you have damage to a cell, there's a number of paracrine signals that exist at the site of the damage. That allows the blood to clot only in that location. If this was not a localized thing and it didn't matter if one little paracrine ion was floating around and you got a chemical reaction to occur, man, that would really stink. In terms of blood clotting, you'd have blood clots all over the damn place. So a paracrine signal only works when it's in greatest concentration from the cell that secreted it. Okay? That's where it's going to have its most positive effect. As it gets diluted down from all the water and very few of them around, it does not elicit a response. All right. So from a blood clotting standpoint, super important. All right. Paracrine signals are going to be also associated with the immune response. You would think that would behoove that one little messenger ion to alert all the other cells that it's passing by. But that's not how that works. All right. We have a number of other cells that are going to do that. Cells. Paracrine signal, it just tells the adjacent ones to start creating cytokines to mount an immune response, because you're going to be infected here in a few days. So paracrine signaling from an immune standpoint is really only effective, again, the same way. When it comes to these types of cell signaling, we can also have synaptic signaling. Synaptic signaling. Okay. This is where we have a specific receptor on a specific target, giving a specific event. Okay. Neurons. Neurotransmitter gets released from the synaptic knob, diffuses across the synaptic cleft, and binds to receptors on the next neuron, on the dendrite. Synaptic knob of a somatic motor neuron releases a specific neurotransmitter, acetylcholine, travels across the synapse and binds to receptors on the motor end plate. And we have a specific event, contraction of that skeletal muscle. So when it comes to synaptic signaling, it's usually specific. specific shape receptor for a specific shape molecule. Okay. Now, I get it. Okay. You can do drugs, similar shaped, the chemical within them to the receptors. Okay, and they can elicit a response. Okay, so again, it's specific, but it's, again, remember, specific shape. Endocrine. Endocrine does the same thing as well. Okay, it will dump its contents into the blood, and as whoever has this receptor on its surface, it's going to bind with it and cause magic to happen. Okay. Thyroid hormone, great example. You have receptors for thyroid hormone all over the place. You have T3 receptors, T4 receptors on every single cell. Every single tissue has one. Thyroid hormone is going to be involved with stimulating growth, basically, affecting metabolism. So thyroid hormone, you'll have this on every single, and so multiple target cells, good gravy. Heck yeah, it does. So endocrine signaling is much more kind of mass kind of communication type of thing. And I mean, paracrine stays localized. Synaptic neuron goes to a specific effector. Muscle, gland, whatever. Endocrine gland, endocrine synaptic communication, endocrine signaling, whatever you want to call it. Much more mass in its terms. Okay, much more mass in its ability to communicate and get things to do things. Get cells to do stuff. Grow. Again, specific receptor. Okay, are really, some things can be brought across. Some things can easily diffuse. Some things need a slight little channel to bring it across because it is too big. But I mean, good Lord. I mean, we just talked about mechanisms to bring stuff across. And if it's polarized or non-polarized, it can either diffuse, non-polar, or if it's polarized or huge, it needs a protein carrier. So there you go. We do have this concept of secondary messenger systems. Secondary messenger systems are wonderful. They're fantastic. Okay. For all of these mechanisms that we've kind of been describing here, we have a receptor on the surface. It binds to it. Magic happens. I have an effect. A secretion. That would be an action. I have sodium channels opening up. That's an action. Sometimes we have a messenger signal bind to a receptor, and then it has an action, but its action is to bind to another receptor inside the cell. And then it's that second receptor that causes magic to happen. Those are referred to as secondary messenger systems. Okay. There are a lot of these within the body. Okay. Calcium can be considered a secondary messenger. Where's my thing? Cyclic AMP is another one. Okay. Tyrosine kinase. There are a lot of them. All right. We are only going to talk about, well, besides from calcium, we're only going to talk about one major secondary messenger system, and that is cyclic AMP. The book goes into all of them: tyrosine kinase, inositol, ATP can be a secondary messenger for some cells. There are a number of them. I think the book goes into them, the vast majority of them. The previous textbook, the Derrickson text, it goes into every single one. You'll go cross-eyed reading them all. The only one that we're going to focus on is cyclic AMP because it's just the most common one. And if you understand that one, then you can figure out all the other ones, to be honest. Okay. Secondary messenger systems, again, we have receptor on the outside, triggers something inside, a secondary messenger out here in the cytoplasm. Okay. It could be outside here in the cytoplasm. It could be inside the nucleus even. Hell, it doesn't matter. But a secondary messenger system can elicit a whole number of various different responses. Okay. One of the major functions of a secondary messenger system is that it helps us to do that thing that I made reference to earlier, upregulation and downregulation. Okay. And these are a couple important concepts here. And I have a picture, I believe, on the next slide. The picture is not from this textbook. Okay. It's from another text. But it does a super fantastic job of giving you a visual representation of what in the heck is going on here with these two things. Okay. Remember, the more receptors we have, the greater the response, the faster the response. So a cell can upregulate or downregulate depending upon different situations. All right. When we upregulate, we increase receptors. Okay. Messenger concentrations are low. There's not enough receptors in the surface of the plasma membrane. This makes it much more sensitive to the messenger. Okay. This could be because there's just a deficiency of an extracellular messenger. It could be a deficiency in the number of receptors on the target. Either way, okay, we package these things up into a tiny little vesicle, transport them to the plasma membrane. V snares and T snares interact with each other and yada, yada, yada. I've upregulated. I have increased the number of receptors on the surface of the plasma membrane. So now it's much more sensitive. It's much more sensitive because I just have more damn receptors there. Okay? When concentrations are really low, a lot of cells will increase the number of receptors on their membrane. This just increases the sensitivity of them. You can downregulate, and it's the exact opposite. If the extracellular messenger is present in excess, it will remove some of these. If the target cell has a huge number of receptors, it will just remove them off of the surface. Okay? And it makes cells less sensitive. Okay? I'm decreasing the receptors on the surface. Okay? And there's some terms here that, we're not getting into it all. As you can kind of see, clarithion-coated pit. Clarithion is just the protein on the underside surface of the plasma membrane. That just tells you the specific location where I'm going to put the receptors. Okay. You can see here we endocytose. I have exocytosed over here. I have endocytosed over here. And I've removed them off of the surface of the plasma membrane. So I've downregulated this sucker. Well, the great thing about this, guys, these little protein markers that tell the receptors where to be, they're just shed and they get transported back to the plasma membrane. Okay. And you can kind of see, we can do a number of things with this. We can store them into some kind of transport vesicle and leave them there. We can break them up and reuse the parts into a different part of the cell. Okay, so we could do a number of things with these receptors that we've downregulated and brought in. Okay. You can also see it's going to help, with some magic inside the cell. So there's some ligand that's bound to the receptor. I can bring it in, destroy the receptor, free the ligand, and the ligand can cause things to happen inside of a cell. So there's a lot of things that can happen when we downregulate. Okay. So these are two major things that are a byproduct of this secondary messenger system. Okay. Because those receptors are inside the cell, in a vesicle. Okay. Receptor binds up onto the surface here, can be brought into the cell, and it says, you need to go to the surface. Well, now it'll go and upregulate. Here is our major secondary system, secondary messenger system right here. Okay. It is cyclic AMP. What does cyclic AMP do? Cyclic AMP do right here. Cyclic AMP activates enzymes. What's enzymes do? Facilitate chemical reactions. That's a pretty broad term, right? And that's the idea. Cyclic AMP can do a number of various different things. Okay. To get this secondary messenger to happen, we need to have a binding on the surface of the plasma membrane first. Okay. Once we get that binding on the surface, now cyclic AMP can be activated inside the cell. And we have a vast number of responses that cyclic AMP does. Okay. It just activates various different enzymes to either add a phosphate, dephosphorylate, can cause some messages inside the nucleus to start reading DNA. Really, the world is our oyster in terms of cyclic AMP and what it can do. It can do a lot of different things. It activates enzymes inside the cell. And now the cell can start doing things. So if cyclic AMP is the second messenger, who in the hell is the first messenger? This is the first messenger. G protein. Please highlight this slide. Okay. G proteins are the first receptor on the surface. G proteins get bound to and then kickstart the cascade of events that occur deep inside the cell for that secondary messenger to happen. So it's these guys that kickstart it all off. G proteins, guys, have three parts: an alpha and a beta and a gamma. Be aware of it because we're going to talk about G proteins later. And there are a number of G protein coupled receptors that are going to be involved with a lot of different stuff in the nervous system and throughout the entire body. Different subunits cause different magic to happen. Okay. Here's my G protein coupled receptor. There's my alpha, beta, my gamma. Here comes a ligand that binds to the receptor here. As soon as that happens, okay, depending upon the cell, that G protein structure disassociates. Okay, and as you can see, there's a couple different ways that it can go. It could be the alpha that goes and binds to the secondary effector. Well, I guess I should say the alpha unit could go and bind to the effector. That kickstarts a secondary messenger system, cyclic AMP. It could be the beta and the gamma subunit that bind to the effector protein and kickstart the secondary messenger system. So there is some subtle differences between alpha, beta, gamma, and what they cause or elicit inside of a cell. And we may hint and reference that a little bit later because it'll come to root and reference itself within the nervous system. But just understanding this picture right here would be fantastic. There is our chemical messenger up there. It's water soluble. Okay. This is cyclic AMP. Water soluble messenger goes and binds to a G protein coupled receptor. There's my alpha, beta, gamma subunit. It disassociates and travels across. Let's just say it's the beta gamma. Okay. Disassociates, beta gamma travels across and activates a secondary receptor, an effector protein in the surface of that plasma membrane. This is what activates, ta-da, cyclic AMP. Cyclic AMP goes and makes enzymes. And that's what this is right here. That is an enzyme. Okay. What does that enzyme do? I don't know. It does a number of things. All right. It can cause proteins to be activated. It can cause phosphorylation to occur. And I can make energy to cause an event to be happening inside the cell. It does a number of things. But cyclic AMP activates enzymes. And those enzymes can do a number of various different things. Yada, yada, yada. What does that mean? It can elicit a specific cellular response. What if it's lipid soluble? Because we have water soluble stuff here in this picture. Do we have lipid soluble messengers? Of course we do. That's a different secondary messenger system. But if I was just
would it still
go ahead.
Would it still go through the G protein?
Different. Slightly different. Okay. It'll diffuse through the plasma membrane, right? Because it's lipid soluble. If it's a lipid soluble mechanism, it can still get different types of G protein coupled receptor activity, but it's not going to be in the plasma membrane. Where's it going to be? Where's another phospholipid bilayer inside of the cell? An organelle, an important organelle involved for directing cellular function.
Nucleus?
Beautiful. Bingo. What's inside the nucleus?
DNA.
DNA. So we have lipid-soluble messengers that can just diffuse through the plasma membrane. They will go and attach to receptors on the surface of the nuclear membrane, and they can get a multitude of things to happen inside the nucleus. The most common thing for those guys, it causes the reading of DNA. And if I read DNA, what am I ultimately going to make inside of a cell?
Proteins.
Proteins. So lipid-soluble messengers, they're slightly different in their secondary messenger system. Okay. And that's where you get the enormous variety that exhibits within the cell and the explanations within all these textbooks that we have for them. Okay. The vast majority of things that we are looking at, processes that we are looking at inside of the cell are going to be the activation of various different things happening outside in the cytoplasm. So I am a hell of a lot more worried about understanding cyclic AMP's use with what it does inside of the cell. Cyclic AMP is a secondary messenger that activates enzymes. Very general. Different cells have different enzymes within them, which is going to give you a different protein activation, ultimately a various different cellular response. Lipid-soluble messengers generally go right to the nucleus and cause the reading of DNA, which is transcription. And then we translate that into a protein, and usually it's the making of a protein. And what does a protein do? Good gravy. It has a bunch of different functions, right? So it can do a whole host of number of things as well. But for us and what we're talking about here, this is the pathway that we're predominantly going to be looking at. Activating proteins to get muscles to get actin and myosin to interact with each other in smooth muscle. Okay. I need this pathway. If I don't have that, smooth muscle doesn't interact with each other. Okay. I don't get myosin kinase activated, and I don't get actin and myosin to interact. Smooth muscle won't contract. So we will see cyclic AMP when we talk about smooth muscle contraction. Okay. So this G-coupled protein receptor, GPCR, that's usually how it's shorthanded, GPCR. G protein coupled receptor is going to be very important for the functionality of a lot of different things inside the body. The major one, smooth muscle. Okay. Secondary messenger system. We have a receptor on the outside. We have another receptor that causes the formation of cyclic AMP. Okay. Our primary secondary messenger. That's the dude. That's the messenger. Okay. And again, it's the second. Okay. Here's number one, my G protein coupled receptor, my GPCR. It binds to the receptor. Alpha, beta, gamma disassociates from the GPCR, travels through the plasma membrane and binds to another effector. Adenylate cyclase is the name of the specific protein enzyme here embedded in the plasma membrane. Phosphorylates cyclic AMP. Cyclic AMP goes and makes an enzyme. And then that enzyme causes a specific event to occur, a specific cellular response. We'll see some of this in the nervous system. We'll see some of it in the muscular system. We'll see some of them all over the body. So we're never, unfortunately, not going to talk about secondary messenger systems and G-coupled protein receptors.